Good morning, everybody. Thank you for being here. Thank you, Monty and the organizing group for the Layman Conference for inviting me to give this presentation, or insisting that I give this presentation. As Monty mentioned, I'm not a DVM. I'm a PhD microbiologist. So when I first started coming to these meetings, I got very confused about things like guilt and GDU and all in and all out. And so I would have appreciated having a glossary like this. And so take a look at it. This is your homework, and but I'll explain these terms as we go through my presentation. This is on Streptococcus suis. Also, as Monty mentioned, my area of research is enteric disease, but recently, in the last couple of years, I got involved in a Streptococcus suis project, and it kind of grew from there. And so I'm moving from the tail end of the pig to the snout of the pig, but only as a sideline. I'm still working on enteric disease. It's fitting that this is the Pijuan lecture, because Carlos Pijuan did the groundbreaking work on Streptococcus suis in the 1980s and in the 1990s, along with Monty and a lot of other graduate students. And since then, Strep suis has always been an issue, but hasn't risen to the top of the issues for swine production. And so there wasn't a lot of research in that area until just recently. So for this talk, I'm just the messenger. There's a group of us involved, most notably April Estrada. She's the graduate student that did most of the work that I'm going to share with you. This is April Estrada. She just finished her preliminary orals and written exam, so she's much happier now. Douglas Marthaler, who used to be at the university and then went to Kansas State and then moved on from there. He is the one that actually dragged me into this project and then left. And he got the initial funding, and so I need to acknowledge him definitely. Marcello Gottschalk is the eminent researcher in Streptococcus suis work at the University of Montreal, and he's pretty much held our hand in helping us interpret some of our data along the way. I also need to give a shout out to Stephanie Rossa at the university and Laura Bruner at the Swine Vet Center. They're the ones that brought this to our attention, the fact that Strep suis was an increasingly important, persistent disease in the last 3 to 5 years. And then, of course, our bacteriology group, both at Kansas State and at the University of Minnesota, that collected all these samples that we used in our research, and Lacey Marshall-Lund for keeping us all organized and on task. And then hopefully there'll be time, because we also partnered more recently with the veterinarians at Abilene Animal Hospital, and we're able to apply some of the tools that I'm going to describe to you in a small, ongoing longitudinal case study. And it's fitting that this is the Carlos Pijuan lecture. As I mentioned, he and his graduate students laid the groundwork for this area of research many years ago, and there's not a lot that's changed since then. So, Carlos, I think you all either know him or know of him, and he was a highly respected researcher. He also passed from pancreatic cancer far too early. He was a professor at the University of Minnesota, the College of Veterinary Medicine, and he also was the founder and director of the Swine Disease Eradication Center. Carlos had a lot of graduate students from all over the world, mostly the Latin countries, and so he had a big influence on research and swine production worldwide. I was not one of his graduate students, but he was very supportive of me. He had a great deal to do with molding my professional advancement. I worked on the tail end of the pig, he worked on the front of the pig, and he was very supportive of everything that I did. He was an avid photographer, took a lot of pictures. He was an internationally recognized scientist, especially, as we mentioned, in swine respiratory disease. He studied the dynamics of infection in different production systems of a lot of respiratory pathogens like H. suis, Mycoplasma, especially PRRS, and of course, Strep suis. Strep suis, it is becoming a more significant swine pathogen, I'm told, from the field the last few years. Causes economic losses. There's humane concerns because of the clinical signs associated with this disease. But the challenge in studying this disease is the fact that there's lots of different strains involved, and they don't all cause disease. The strains that cause disease, the systemic strains, they do cause meningitis, arthritis, endocarditis, epicarditis, polystyrocytis, I don't think my pointer works, and septicemia. And then there's opportunistic Streptococcus suis, and those usually only cause disease as a result of co-infections with especially PRRS and influenza virus, and with bacteria like Bordetella and Pasteurella. And then there's a whole bunch of commensal Strep suis that are actually normal inhabitants. They colonize early in piglets and generally are just commensals and don't cause disease. So we need to sort through all of these subtypes, I'll call them, of Streptococcus suis in order to find the true pathogen. So how do we diagnose Strep suis disease? Preliminary is by history and clinical signs, age of the pig, gross lesions. You all know that better than I do. And confirmatory is where my laboratory comes in. The histopathology combined with culture of the organism is very easy to culture, and I never thought I'd be standing up there saying that about a bacterium. It's very easy to culture. Dr. Rossau asked me to remind you, though, to always send in brain samples if you can, because that is the best tissue for culture. Other systemic tissues will work as well. The problem is culturing and speciation is not enough. You need to subtype the organism. You need to see whether it's one of those systemic, opportunistic, or commensal strains. And on top of that, there's a lot of different strains that are involved in the disease, and whether or not they're all causing the clinical signs or just certain few still needs to be figured out. So, as I said, there's not a lot of information recently in the United States. There's quite a lot of information coming out of Asia and Europe and Canada, but the last publication looking at the distribution of Streptococcus in the United States was about 10 years ago. And over those years, too, only serotype 2 was the one that was commonly studied. That is a known virulent strain, pathogenic strain. And so most of the studies are based on that. But as you'll see in a little bit, serotype 2 isn't really the problem here in the United States recently. Why, I don't know. Maybe we've done a good job of vaccinating serotype 2 away, and a few other serotypes have taken up that niche. So then, and complicating it even further is the fact that it's not just the bacterium that causes the disease. There's impact of the environment and the pig itself as well that have roles in the severity of Strep suis disease. How do we control the disease? There are, you probably know this better than I do, antimicrobials work. They work pretty well, but you have to get them in early. Early intervention is key. And then there's the trend towards reducing the use of antimicrobials in animals. And then another problem is if you treat the disease away, then you don't get the natural resultant immune response. So vaccines look like they may be a better option, but there's no commercial vaccines regularly available, I guess, here in the United States. And a lot of the recent research in Canada shows that the vaccines are not cross-protective. So this makes isolate selection really important, and but it's complicated, as I mentioned. Also, the best approach for vaccinating is not very clear. Do you vaccinate the piglet? Do you vaccinate the sow? Or do you vaccinate both? And the efficacy of these vaccines is difficult to measure. So how did I get involved? Well, the sequence of events is, well, it started way back when I saw this video that's gone around of the classic signs of the disease of piglet paddling, and that's the classic neurological sign of strep suis infection. And I'm not going to show you the video because A, I'm not very good with the technology to make a video work, and B, it's really heartbreaking to watch. But this video has really stuck with me, and so when we got the opportunity, recently when some of the clients like Swine Vet Center, Laura Bruner, brought the information to our diagnostic lab that, hey, we're seeing a lot of strep suis out there and it's persistent and we're not able to control it. And our VDL at the University of Minnesota, the pathologist, confirmed this. And then shortly after that, the Swine Health Information Center listed their new bacterial disease matrix, the list of bacterial diseases that are of importance or going to be of importance, and strep suis topped that list. And it ticks all the boxes in that list. It's a zoonotic disease, and I won't even get into that, but it's becoming increasingly important in Southeast Asia and actually does cause quite a bit of fatalities. It has large economic impact. It has an impact on the health and welfare of the pig. There's not very good control methods. And finally, there's inadequate diagnostic methods. So that's where we come in, the inadequate diagnostic methods. So what we do, I work with the diagnostic lab and we develop new diagnostics. And hopefully we can help then identify approaches for intervention based on those new antibiotics. And so I'm going to tell you a little bit about some of the new tools we've developed for addressing strep suis disease. And then we had the opportunity to partner with Abilene Animal Hospital. They had a persistent problem with strep suis just recently, and they approached us, and we used this longitudinal study in order to investigate the utility of some of the tools that we've developed. So if there's time, I'd like to go through some of that with you as well. So, and as I said, understanding strep suis is going to be very complicated. There's the pig, and it's dependent on their immune response, whether they have other co-infections. It might even have something to do with the microbiome, whether these pigs are susceptible to strep suis, no matter if it's a commensal isolate or a pathogenic isolate. The environment plays a role. We're not exactly sure what factors are the most important. Stress is a big one. Overcrowding, temperature fluctuations, all of the above. Husbandry, antimicrobial use come into play. And then the other factors that are involved are the strain of strep suis, as I mentioned, what virulence-associated genes, VAGs, virulence-associated genes, that strain may have, etc. So my group, since we're bacteriologists, chose to start with Streptococcus suis and specifically to look at the DNA. And the overall strategy is to utilize whole genome sequencing, and we sequenced over 200 strep suis isolates, and we chose these isolates though to represent pathogenic and commensal isolates for the most part. And so what our ultimate goal is to compare the genomes of pathogenic and commensal isolates in order to identify the factors that are involved in pathogenicity of strep suis. And so this is the four aims that we had, and ultimately will be the 4 accomplishments that April Estrada obtained. So the first aim was to characterize the subtype distribution of strep suis in the United States. We didn't really know what it was in the last 10 years. And we need to understand the subtypes that are involved, whether they're subtype by serology or by a genetic method called multi-locus sequence typing, MLST, referred to as sequence types, or ST. We investigated both approaches. And then we assigned pathotypes to our culture collection, to our over 200 isolates, and these pathotypes then will help us identify which subtypes, serotypes, or sequence types are important in Streptococcus suis disease. Then we went on to identify the virulence-associated genes which are in these sets, the pathogenic and non-pathogenic of Streptococcus suis, hopefully so that we can identify a panel of virulence-associated genes that are associated with pathogenicity. And then finally, we used genomic SNP typing to look at differences between individual strains within and between systems or sites. And any of you who have submitted PERS for sequencing and gotten back a dendrogram, it's a similar process as that. It helps you detect and monitor isolates that are within an outbreak and or track outbreaks. So our first objective was to look at the distribution throughout the United States, and we ended up with 200 Strep suis isolates that fit our criteria. They came from the University of Minnesota and Kansas State Diagnostic Laboratories from 2014 to 2017. We made a great effort to collect as many isolates as we could from all of the swine-producing states and cover as many systems as we could and sites without having more than 2 isolates from a certain site. And so in the end, we had isolates from 16 systems and over 70 different sites within the United States. Of course, most of our isolates came from Minnesota, as it was samples, for the most part, that came into the diagnostic lab. The darker blue states are the ones that yielded the most samples. The gray states we did not have samples from. But in the end, we had samples from 20 different states. And spoiler alert is that the predominant serotype was a serotype called 1/2, and that was found in 13 of the states. And serotype 1/2 is actually an individual serotype. It cross-reacts with serotype 1 anisero and with serotype 2 anisero, but it has its own unique set of CPS genes. And this serotype 1/2 was most common in 5 states, and it was the 5 states up and down the Mississippi River. We also, the MLST sequence type 28 was the one that was most common in our set of 208 isolates, and that was found in 8 different states and most common in the same 5 states up and down the Mississippi River. So this is actually all of the information on distribution of Strep suis, and what it shows is along the x-axis. Okay. Along the x-axis, we have the serotype, and there were 20 serotypes in all that we looked at. And along the y-axis is the frequency of isolation. Along the top is all of the different sequence types that we have that were color-coded. And you don't need to see this. I'll tell you what our take-home message is. Our take-home message is that serotype 1/2 and 7 were the most common serotypes that we found in the United States. We found 58 sequence types. Those are the ones that are listed at the top of the figure. And the most common sequence types that we identified were 28, 94, 1, and 108. Sequence type 1, I wish my poster worked. Sequence type 1 is split between serotype 1 and serotype 2. And also note that serotype 14 contains 100% sequence type 1. Also, sequence type 108 was split equally between serotype 3 and serotype 23. So this tells you that there's a great diversity of Strep suis here in the United States, especially as detected by sequence typing. So then the next thing, well, so what does it matter whether or not these isolates are present or not? Some of them are commensals, some of them are systemic pathogenic isolates. So what we did as we chose our 208 isolates, as we characterized them by pathotype. And we characterized them as 3 different pathotypes, either the pathogenic pathotypes, meaning they came from the systemic tissues, most often the brain or the heart, from pigs that had typical signs of CNS, typical neurological signs of Strep suis infection. Then we had our set of possibly opportunistic that came from the lungs, of pigs that were submitted to the diagnostic lab that clearly did not have neurological or systemic disease. And since these animals came to the diagnostic lab, they likely did have some respiratory disease. And then we had our commensal set, which were the hardest set of all to come up with, and this is a set that we went out in the field and got from sites that had no history or weren't treating for Streptococcus suis. So when we assigned these 3 pathotypes to our set of 208 Streptococcus suis, we could start seeing which ones in the United States are more pathogenic and which ones are more commensal. So this next stack graph shows the association of subtype, the serotype to the pathotype. And so again, on the bottom, we have our 20 serotypes. We have our 20, there we go. We have our 20 serotypes. And then in rectangles, you can see the ones that are most prominent. We have serotype 1, serotype 1/2 again, serotype 2, serotype 7, serotype 14, and serotype 23. The dark red, or the rose-colored, are pathogenic pathotype, and the light pink color is our commensal pathotype, and then the medium pink color is our possibly opportunistic group. So our take-home message from this is that serotypes 1/2, although we didn't have very many of serotype 2, as I mentioned, in our United States set of samples, serotype 7, 14, and 23 were classified as the pathogenic pathotype. That means that proportionally more than 75 or 80% of the isolates that were in that pathotype fit that criteria. And then we had a statistician involved that helped us show that our conclusions were statistically significant, and he had us do odds ratio analysis, and odds ratio analysis supported this conclusion. And then also note that serotypes 1 and 14 are the 2 serotypes that are entirely composed, 100% pathogenic pathotype. And this will come into play later when we talk about this longitudinal study. Same thing with distribution of the Strep suis pathotypes by sequence type. Again, we see the sequence types, 58 of them in total, along the bottom of the figure, and the frequency is on the y-axis. And what you can see here, well, maybe you can't see, but I'm going to tell you, is that sequence type 1 and 28, 94, and 108 were mostly classified as pathogenic. That means like 75 to 80% of the isolates within that serotype were pathogenic. And again, these associations were supported by odds ratio analysis. We also see that sequence type 1, the very first one in the figure, is made up entirely of the pathogenic pathotype. And this will again come into play when we get to the longitudinal study. All of these singletons, many of these singletons in the green circle on the bottom belong to the commensal pathotype, and that's because they haven't been well studied, and so they have new sequence types. Although we have a couple of sequence types, 750 and 821, that are composed of multiple commensals. So I don't expect you to see or understand, but unless you're used to looking at heat maps, but this summarizes everything. So this is a heat map based on the sequence type sequencing, and along the right side you see our 208 Strep suis isolates. On the bottom are 208 Strep suis isolates, and so this shows the relationships between the sequences within those isolates. And the first column on to the right of the heat map is our pathotypes. Dark rose is pathogenic, light pink is commensal. The second column is serotypes, are 20 different serotypes, and the third column is sequence types, are 58 different sequence types. And you don't need to see that, but what we found is that there's 4 main groups or clades in this heat map. The top 3 clades clearly associate with pathotype. This clade, the largest one, the one in the middle, associates with the pathogenic pathotype that's represented by serotype 1/2 and sequence type 28. So that's the majority of the isolates that we saw in the United States. And that's a new finding that hasn't really been reported, that serotype 1/2, sequence type 28 is associated with pathogenicity. This second clade or group, the one in the middle, the small one, is associated with the well-known pathogenic pathotypes, the Strep suis of sequence type 1, serotype 1. Now, normally it's serotype 2, and there are a few serotype 2s in that group, but most of our pathogenic pathotypes were found in serotype 1. And then the last clade, the last group on the top, is made up of mostly commensals and a few opportunistics, and that clade has a variety of serotypes and a majority of novel sequence types. So that clade is essentially our commensal set of pathotypes. The clade on the bottom, the group on the bottom, I'm not really sure what that is going to turn out to be. Possibly our opportunistic group, but there's a lot of pathogenic pathotype in that group as well. So the take-home from this heat map is that if you cluster by sequence type, you see that there's 2 different pathogenic groups of Strep suis, the classic one, that's sequence type 1, and the novel one in the United States that hasn't been well studied, the sequence type 28. And then we see 1 group of commensal pathotype. So we moved on to so we've done the top 2 aims in our little project wheel here. It's supposed to be a large Streptococcus suis with DNA in the middle, my rendition anyway. And I just gave you an overview of what we found, the take-home messages. But this is published. This is April Estrada's first publication, and it just recently came out in the Journal of Clin Micro. So if you want to see even more detail, it's in that publication. The next approach was on the bottom right-hand side, where we used the whole genome sequences that we generated for these 208 isolates to look at the virulence genes that are associated with the pathogenic group versus the commensal group. And I'll just give you a quick overview on this one too, because this is actually the subject of a poster number 46, that's presented here by April Estrada. But what we did in that study, we meaning April, is a little bit of the background is that there's over 100 putative virulence-associated genes for this organism. Not many of them have been proven necessary for virulence. It's kind of a messy story. The classic ones that are considered to be important, and this is just for serotype 2, which we don't see a lot of in the United States, but the basic ones are CPS, capsular proteins, MRP, EPF and SLY, which is the hemolysin. But again, these are only for serotype 2. We don't know what the repertoire of virulence genes is for these other serotypes. So what we did is, for 66 of these 100 virulence-associated genes, we determined the distribution of those genes in our pathotypes of Strep suis. And again, one more heat map. This one's going to be a lot easier now that you've seen the first one. This will be easier. So this heat map, note that the dark blue means that the gene is present, and the light blue means that the gene is absent. And so on the right side, again, are our 208 Strep suis. They're annotated on the far right by pathotype. The bright rose are the pathogenic pathotype, and the light pink are the commensal, and the medium pink are the opportunistics in between. And again, what we see is we see 3 large groups in this heat map. Clusters or clades, whatever you want to call them, and the bottom 3 associated with pathotype. So this one on the bottom, this small group, again associated with our sequence type 1, our classic pathogenic pathotype of Strep suis. The one in the middle also was composed almost entirely of the pathogenic pathotype, and that corresponded for the most part with our sequence type 28, although one small difference when we look at the sequence type association is that there's a variety of serotypes included in each of these groups, but all of them are either sequence type 28 or sequence type 1. And then this third group, the one in the middle, is most of our commensals. And again, the one on the top, we're not sure what it is. We need to do further analyses. Right? So the take-home message there, not ready for that. So on the bottom, you might not be able to see it, but I have 4 arrows. And those 4 arrows represent the 4 variance-associated genes, the classic ones that have been well published: the capsular gene, the MRP, the EF, and the suilysin. Those 4 genes, remember the dark blue is the presence of the gene in our 208 isolates, and the light blue is the absence of the gene. Well, those 4 arrows on the bottom, the first one is the capsular genes, 2 of the capsular genes. They're present in most of the, in this group, the second group from the bottom, our sequence type 28 group, and in about 75% of our sequence type 1 group. The other 3 genes are almost 100% present in our sequence type 1 group, our known pathogenic pathotype, and pretty much absent in all of our sequence type 28 group. So we think that a better place to look for virulence-associated genes are within this green rectangle, because that is where there's several genes that are found in both the sequence type 1 group of genes and the sequence type 28 group of genes, but are almost totally absent in our commensal group of genes. So the take-home there is that this clustering by virulence genes illustrates, again, that there's 2 groups of pathogenic pathotype, and they're represented by sequence type 1 and sequence type 28 here in the United States, and 1 group of commensals. And they correspond pretty nicely with the clustering that we found by MLST typing, sequence typing. And our statistician made sure that we did this statistically instead of visually, and so we did linear regression analysis, lasso analysis is what it's called for short, and identified candidate variance-associated genes that predict these 2 pathotypes. And we did find a group of genes that more than 80% of the time do predict both of these pathogenic pathotypes, and we're still investigating those genes. So the last topic is, of course, we want to know if we can tell isolates apart, whether or not they're in the same sequence type or not. And we're basing this on SNPs, single nucleotide polymorphisms. I think you're used to this if you do PERS testing in dendrograms. And so we took these whole genome sequences and analyzed the nucleotide changes, comparing the pathogenic pathotype with the commensal. And we got this cluster analysis, this tree, we'll call it a tree, of our 208 isolates. And I don't expect you to be able to see it, but you can see that the sequence type 1s, again, cluster up at the top. The sequence type 28s cluster here. And our commensals are all on the bottom, but they're very diverse. They're all over the place. We also found that we color-coded. We had 16 systems that we knew of that these isolates came from, and so we color-coded them by system. A through P, each of the letters was a different color. And we found that sequence type 1 seemed to happen quite sporadically. But when we look more closely at just the section of the tree that shows sequence type 28, you can see that they're almost all identical, the sequence type 28s, and they are collected from 9 different systems in 9 different states. So there's something about sequence type 28 that makes it stay pretty homogeneous. So our conclusions are that we can demonstrate the diversity of the isolates, and we can track and identify individual isolates within a sequence type or a subtype. So, as promised, we're going to talk about some pigs. Oh, we don't have time to talk about pigs. We don't have time to talk about pigs. Okay. I will do my concluding slide. By the way, the good news is this study is going to be presented in its entirety at the Purdue conference coming up by the Abilene group, and so you'll get the true story there. And so my conclusions are that we're now able to pick strains for preventive strategies based on serotype or sequence type. We know which subtypes are associated with pathotype, and so we can develop new diagnostics, maybe direct PCR, to detect the important subtypes. We have a panel of virulence-associated genes that might actually identify virulent isolates directly, and then now we can monitor within and between strains, within and between systems. And with that, this again is my list of acknowledgments. And especially I'd like to acknowledge Carlos Pijuan's contribution to the field, and we're hoping that we'll be able to follow his footsteps. They're quite big.
Thank you.
Thank you all.